Hello everyone. So welcome again to another video. So in this biology lesson that we were going to tackle today, so we will going to discuss one that is related to the DNA and how the DNA creates protein. So basically, what will happen? Alright, so basically in this lesson, we will going to answer what will happen if there is something that intervenes or that that changes the way on how the DNA makes protein in the overall process. All right, so before we have the discussion, so let's take a look this one first. So are you familiar with this scene in a movie of Avengers? So let's play this movie.
Might be a really good time for you to get angry.
That's my secret, Captain. I'm always angry.
Alright, so as you see, Dr. Banner transformed to become an incredible Hulk, or rather Hulk, yan. So as in the process, from a normal human form, Dr. Banner transformed to be an incredible Hulk. So the one that you see there, the big green guy on the movie. Alright, so in this movie, so in this particular scene, the transformation of Dr. Banner to Incredible Hulk is a clear example or a classical example of what they call epigenetics. So what is this epigenetics all about? This is the study of changes in the characteristics of an organism without any changes in its genotype. So meaning to say, in this particular process, what is happening here is the characteristics or the appearance of the organism will be the one changing, but not affecting its whole DNA. Alright, so without changing the genotype. So meaning to say, you will just change the phenotype without changing the phenotype. That is what epigenetics is all about. Now, in this case of Incredible Hulk, so one of the reason why Dr. Banner changes into this form is because of anger. Alright, so meaning to say, the one that triggers the change is the external factor, which is anger. Alright, so in some cases, because of anger, Dr. Banner transformed itself instantly to become Incredible Hulk. Alright, so in reality, these epigenetics things or these things that we have right here on this particular scene on the movie also happening in nature, but not the way it is happening here, which is instant. Alright, so this is a clear example of what they call epigenetics. So since we are talking about genetics here, so the one that I've shown you, alright, so the one that I've shown you on the movie is a clear example or part of our lesson today or part of this video, which is called the mutation and genetic disorder. Alright, so in this topic, we will going to talk about how or what are the different types of mutation and what are this genetic disorder all about. Alright, so basically, what is mutation? Mutation is a change in the DNA sequence that may affect the whole chromosome or just one gene. So it depends. So if the DNA sequence is changed, so basically that is what they call mutation. So any changes in the DNA sequence that you have right here, so the DNA sequence, this one, so any changes in the DNA sequence that can affect the chromosome or even a part of the gene, it is called mutation. Alright, so mutation or any changes. Now, there are two kinds of mutation. So we have chromosomal and gene mutation. So basically, in today's lesson or in today's video, we're going to discover what's the difference between chromosomal and gene mutation and what are the typical example of these mutations that we have right here. Alright. Now, typical causes of mutation is if the DNA fails to copy accurately during cell division, especially during meiosis or somehow in mitosis. Alright, so if the DNA fails to copy accurately the way that it is usually copied during replication, meaning to say the mutation process or it is one of the causes of mutation. Not only during the replication process, but also during the protein synthesis process. If the mRNA or if the RNA didn't copy accurately the DNA, it will cause certain changes in the organism. Next one. Aside from DNA fails to copy accurately, external influences can be attributed to the causes of mutation. So what are these external influences? One is chemicals. So there are certain chemicals that can cause mutation, and another one is radiation, and the other one is infectious agent. So let's start first with the chemicals. So there are some chemicals that trigger changes in the cells, like there are some chemicals that changes the amino acid group to become the different group. All right, so there are chemicals that speed up the amination of this particular nitrogenous bases. So chemicals, or these chemicals are also known as mutagens. So these mutagens, not only chemicals but also radiation, contributes to the changes of the organism. Thus, the organism tends to mutate. Now, in terms of radiation, now if you recall one lesson in physics, which is the electromagnetic spectrum, all spectrum, or all part of the spectrum which has a higher frequency, from starting from the ultraviolet down to the UV rays, down to the we have the gamma rays. These are high frequency radiation. So it means that this radiation is said to be ionizing radiation. When we say ionizing radiation, this radiation has tends to rip off the DNA molecules. So these molecules that are found in the DNA tends to be separated, tend to disintegrate with whenever this ionizing radiation is present. So we call this radiation as ionizing radiation because it has high frequency and high energy radiation. Next one, there are some infectious agents like viruses that can cause mutation. Alright, so these are retroviruses like the HIV and others. Other examples of viruses like polio or something like that. Alright, so these viruses tends to change the composition or DNA composition of their host target. Alright, so basically these are the two causes of mutation. One, DNA fails to copy accurately, and external influences. Alright, next one. Now, the first part of this discussion, which is called the types or kinds of mutation, one of those is the chromosomal mutation. So what do we mean by chromosomal mutation? This is an unpredictable change in the chromosome. So again, there's a word change because of the word mutation. So if there's a changes in the chromosome, then it is attributed to be related to chromosomal mutation. Next one. Alright, so these changes are often brought on by problems during meiosis. So if there's a problem during meiosis, so in which the chromosome inside the nucleus tends to divide, so if there's a problem with that particular cell division, so it will affect the total number of chromosomes present on an organism. Or there are some cases by chemicals, it can cause some chemicals and some viruses can cause certain effects in this particular chromosome that changes its structure and how it functions. Next one. Next one is chromosomal mutation is a result or can result in changes in the number of chromosomes. So as I've said earlier, so not only the changes in number of chromosomes but also changes in the structure of the whole chromosome. Alright, so that is chromosomal mutation. Whereas, so if you take a look at this, so this is a typical example of a normal chromosome. So any changes in the structure and the number of chromosomes that we have right here, if this is human, it should be 46. So if there's a changes or in the number of chromosomes that we have right here, therefore it is called chromosomal mutation. Meanwhile, The gene mutation is somehow a different kind of mutation. It involves the DNA sequence. Any changes, so the word here that we use is alteration. So it is attributed to mutation. So any changes in the DNA sequence that makes up that particular gene. So changes in that DNA sequence is it is called gene mutation. So the gene that creates this protein needed by our body, or needed to form an organ, a body part, or whatsoever, to make the body part works perfectly. So if this gene is changed or altered, therefore it can cause mutation to an organism. Alright, so there are two kinds, or there are two classification of gene mutation. One of those is hereditary mutation. So when we talk about hereditary mutation, this is a type of mutation which is passed from parent to offspring, or also known as germline. It is also known as germline mutation. So why germline mutation? The germline here that we are talking about is the sperm and then the egg cell. So if the sperm and egg cells have this certain mutation into their nucleus or into their DNA, then therefore this kind of, or this mutation will be passed down to the offspring, and the offspring will manifest the same mutation that is found on the parent. So that is why it is called hereditary mutation. And not only on the offspring, but for the next generations of that particular organism. Alright, next one. The second kind, or second classification of gene mutation, is called the acquired or somatic mutation. So what happens here in the acquired or somatic mutation? From the word itself, acquired, it means it is obtained. Alright, it is obtained. So in what way that the person or that the organism obtain this kind of mutation? Now, some of these are caused by environmental factors like radiation, alright, or any mistakes that happens during the cell division. Alright, so this particular mutation can change the organism drastically. Yan. So, and it is also attributed, so this kind of mutation is also attributed to the next topic that we will going to have in the future, which is called evolution. Alright, so again, this the two classifications of gene mutation. We have hereditary and acquired mutation. Alright, so okay, so let's start first with the first example of gene mutation. The first example here with the gene mutation, so the one that we are talking right here, is a gene mutation. So this is what they call deletion. Alright, so deletion, it occurs when one of the part of the chromosome is left out. So meaning to say, if one part of the chromosome is deleted, so this is before, and after deletion, so what happens is it completely changes the overall structure of the chromosome. Alright, so what you see here, the D is deleted, then therefore it changes the chromosome. So aside from that, deletion is also it is also an example of chromosomal mutation. Alright, next one. Now, this is the gene mutation. So if one letter from a codon is deleted, it will affect how the codon is translated. So what you have right here in the first sequence that we have, alright, so we have before and after mutation. So what happens here is the nitrogenous base A is deleted in this process. So what happens, from the word or from the codon cat, CAT, it changed to become CTC. Alright, so the CTC codon has different amino acid coding. So from histidine, it will change to leucine, and other codons which is next to the codon which is affected by the deletion is also changed as well. So from histidine, it becomes isoleucine respectively. So now, is there an effect on the organism only by changing the amino acid? Yes. Alright, so if this mutation happens on a neuron, alright, developing neuron, so therefore that neuron's function will be compromised. Alright, so Alright, one example of deletion that we have right here is the deletion of chromosome number 5. So, as you can see here, all chromosomes are normal except for number 5, in which there is a part of the chromosome here that is deleted. Alright, normally, it should have equal size with its partner chromosome, which is called the homologous chromosome. Alright, so it should be the same, but because of deletion, it becomes different from its partner chromosome. Alright, so what is this disease? The deletion of number 5 chromosome is called the disease called is Klinefelter Syndrome. Alright, so it is spelled as Klinefelter syndrome. Alright, so in which the individual which has this kind of this syndrome has the voice box is not functioning well the way it should be. So that's the reason why every individual that has this syndrome, it cries like a cat. So if you happen to hear a cat's cry, so that is how an individual which has this syndrome somehow cries. Okay, so that is Klinefelter Syndrome. Another one is cystic fibrosis. Now this is a disease which leads to the amount or inappropriate or development or accumulation of water or accumulation of fluid into the lungs without any reasons at all. So when that fluid accumulate in the lungs, so what happens is it will cause death of the particular individual. Alright, so that is cystic fibrosis. So as you can see in this image, the lungs already filled with this fluid, so which is created by that changes only in the chromosome of this particular individual. Alright, so you see the effect. Next one. Next kind of mutation is called insertion. Again, insertion can be also called as part of chromosomal mutation. Alright, so insertion in this way, as you can see here, this is the normal chromosome. So what happens here is there is another set of parts from another chromosome which is inserted to this chromosome. That's the reason why it changes its overall structure. Alright, so that's the reason why this chromosome, this one over here at the bottom, is different from the chromosome on top. Alright, so that's how insertion happens. So when this one is deleted and reinserted, or it is inserted to another chromosome, so insertion happens. Now in gene level, what do we have right here is before and after the. Alright, so this is before and after the insertion. So what happens here? One nitrogenous base is inserted along or in between the sequence. So, is there an effect on that? Yes, there is an effect on that. The effect of that is it will totally change the amino acid that normally formed by that particular sequence. So, only by inserting this A nitrogenous base will change totally what is coded by that particular sequence of the DNA. Alright, so threonine, and we have serine and so on and so forth. Alright, so instead of histidine all throughout. Alright, so one of the example here is the Huntington's disease. This is a gene mutation in which the genes that makes the neurons. Alright, so the genes that makes the neurons function properly is affected. Alright, so the person that you see on the picture is affected by the Huntington's disease. So its neuron or his neurons are affected. So it doesn't develop or it doesn't function normally. So there is a severe mental retardation and the cognitive function of the brain is also affected. Alright, so that's the Huntington's disease. Alright, so another example of chromosomal mutation and also gene mutation is inversion. So what happens here? This is the normal chromosome. So we have here the normal chromosome. Now during the inversion, it is, or the two portion of the chromosome is breaks out, and it is reinserted backwards in opposite way. So this is the normal, which is A, B, C, D, E, and during the inversion, it is reinserted inversely. Alright, hence it is called inversion. So what happens, instead of A, B, C, it is now A, D, C, B, E. Alright, so is there an effect on the organism whenever inversion happens? Yes, of course, there is. Alright, so one of the effects here, or one of the notable disease that is caused by inversion is hemophilia. Alright, so hemophilia is caused by inversion of a certain gene that are found, or that is found in a particular chromosome, and it is reinserted backwards, the one that we have here in the picture. So instead of A, B, C, D, E, F, it becomes A, B, C, F, E, D. So it is reinserted backward. Then if this one, alright, so if this one is for the development of protein that enable us to form blood clot, then therefore that particular ability will be affected if there is an inversion that happens on that particular gene or chromosome. So technically, hemophilia, those people who has hemophilia they have inability to form blood clot after they have a certain wound, or certain blood, or certain scratches on their body. So inability to form blood clot. All right, next one. Next is the translocation. So in translocation, this is similar to deletion and insertion. All right, so somehow they are similar, but in translocation, what happens here is when part of the chromosome breaks off and then added to different chromosome, this is where the translocation happens. So as you can see here, the normally this chromosome looks like this one, the one on top. Now after translocation, this WX is deleted and reinserted two other chromosomes in the sequence, all right, or in the set of chromosomes that present on that person's body. All right, so as you can see, the WX is now inserted on WX ABCDE. So what happens on this chromosome? It is now completely different. It is now completely different on its structure, not similar to the one on top. All right, so that is translocation. Now if you take a look at this picture, you can see how translocation occurs in a chromosome. So as you can see, this chromosome should be red. So there's one part of the chromosome here which is odd, which is color blue. So this color blue, or this blue part that we have right here, came from the 22nd chromosome over here. And also the part of this red chromosome is transferred to the 22nd part. So if this happen, there will be an effect on the organism. Alright, so one example of a syndrome that is attributed to translocation is the Albinism syndrome. So what happens here is the affected chromosome, or the affected gene here, is the gene for the development of a normal functioning of the liver. So what happens here is, because the translocation affects this particular part of the gene that functions for the liver, so the liver will not function properly. So there is now an accumulation of different substances that are present on the liver of these people in the picture. So they develop differently. They have different skin. Yan. So they have different skin, or different characteristics compared to a normal person. Next one is the Burkitt lymphoma. So this is another example of translocation, in which what happens here is there is some part of the chromosome that is translocated, and as you can see here, so it is translocated. So from this chromosome, it is translocated to the other part, and what happens here Because of the translocation, the lymph nodes function is compromised. Alright, so sometimes this functioning of the lymph nodes, because they are not functioning very well, it can lead to cancer. So the one on the picture has a certain disease called Burkitt lymphoma. So it affects how the lymph nodes function normally. Alright, next one. Alright, so there's another kind of mutation here. It is called missense mutation. So what happened here is, this is a kind of mutation that changes one base of the DNA or one DNA base pair that results in the substitution of one amino acid for another in the protein made by that particular gene. So this is a gene mutation. So what happens here is, alright, so there's no deletion. Alright, there's no deletion, there's no insertion happened, only by changing that nucleic rather nitrogenous bases from A to C. So we don't know where this A came or where this C came from. So only by changing the A to C, it changes the amino acid that is formed by the DNA sequence. Now, unlike inversion, deletion, and insertion, which affects the other amino acid, in this mutation, it only affects one set of codon and one amino acid, so that the codon should produce. So instead of histidine, it will become proline. So what's the reason? You change the A nitrogenous base to become a C nitrogenous base. Alright, so that is the missense mutation. So in evolution, in evolutionary path or evolutionary changes in an organism, missense mutation is somewhat seen on this process. Alright, so in evolutionary changes. Alright, so one example of missense mutation is the sickle cell anemia, so in which the blood cells are shaped like a crescent moon. Normally the blood cells should shape like a circle. Alright, so a circle that we have right here. But if you have this kind of mutation, what happens is the cell will become sickle shaped. Alright, so next one. Tay-Sachs disease. So there's a mutation that happens in the nervous system of this person in the picture. So it affects the cognitive and normal function of the nervous system in total. Alright, so that's the Tay-Sachs disease. Alright, next one. Alright, next type of mutation or next example of mutation that we have right here under the gene mutation is called as nonsense mutation. So why is it nonsense? Because from the definition itself, the altered DNA sequence is prematurely signals to stop building protein. So meaning to say that the protein synthesis process is somewhat prematurely stopped. Remember our lesson with protein synthesis. There is a start codon and there is a stop codon. So there's a start of the protein synthesis, and the protein synthesis will not continue forever. So it should stop at a stop codon. Alright, so in this case, it started normally, but it ended prematurely. So that is the nonsense mutation. So if you take a look at the gene sequence of this mutation, you can see, alright, so you can see that this mutation, if you will going to code this letter, alright, if you will going to code this, or if you will try to look at the amino acid that is coded for this codon, so we have here U. Alright, so U, or rather A, U, C. Alright, so we have here the A, U, C. Alright, so what happens here is this is a stop codon. Alright, so this is a stop codon. So basically, instead of moving forward, instead of continuing the protein synthesis, it is stopped prematurely. So meaning to say the whole process, the whole process here will stop, and then the overall amino acid sequence will also be changed. Alright, so now, next one. Next type of mutation is the frameshift mutation. So from the definition itself, it occurs when addition or loss of DNA bases changes the gene's reading frame. So this is like this is like a type of this is a type of mutation in which there is the RNA which copies the DNA sequence tends to read the DNA sequence improperly. All right, so the RNA polymerase didn't or forgot to read one nitrogenous base, thus it will change the protein that is coded by the DNA itself. Alright, so this is how it looks like. So in this case, this is the normal set or normal pattern or normal arrangement of the amino acid. Because of error in reading, alright, so the RNA polymerase forgot to read this particular nitrogenous base. The reading frame is now affected in total. Alright, so instead of having histidine, serine, histidine, valine, so on and so forth, it become isoleucine, histidine, rheonine, tyrosine, serine, cysteine, and so on and so forth. Alright, so it is now completely changed because of this error in reading of this nitrogenous base that we have right here. So that is the frameshift mutation. Next one, the repeated expansion mutation is a type of mutation in which the DNA sequence is somehow repeated over and over again. Or sometimes it is repeated in a particular row of that DNA sequence. Alright, so here's how it's done. Okay, so this codon that we have right here is repeated again on the next one and on the next one. Alright, sometimes it is in some cases it is longer than that we have right here. Alright, so the CAG here is repeated again and again. So what happens here is the amino acid that is coded that should be next, that valine should be next, is now inserted by new amino acid coming from this set of codons that you have right here. So will this have a drastic effect on the organism? Yes, it is. So please also take note that any mutation that we discuss right here, there will be a changes in the organism. It might be an appearance, or it might be a physical appearance on the organism, or it might be how, or it can be on how the cells in the body of that individual will function normally. Alright, so like the neurons, the cells in the digestive system, so on and so forth. So if mutation happens on those cells, technically the functioning of that particular cell will also be compromised. Alright, next one is duplication. Alright, it is similar to non-repeat, or it is similar to repeat expansion mutation, but the duplication sometimes, alright, this duplication process happens in the chromosome level. Alright, so what happens here is there is a part of the chromosome that is duplicated unnecessarily. So what happens, the section of the DNA in this part of the chromosome is also altered. So what it means that if this is an organism, so it might be the physical appearance or the functioning of the cell that will be compromised by this kind of mutation. Alright, so thus that this is what they call the duplication mutation. Alright, so one of the example of duplication mutation is the Charcot-Marie tooth disease. So in which the person who has, or an individual who has this kind of disease, has inability to control the muscle movement in the feet and in the hands. Alright, so as you can see, this person cannot control properly the muscles in this region of his body. Alright, so that is one example of duplication in the chromosome. Alright, now those were some examples of gene mutation and as well as chromosomal mutation. Alright, now in this section of our video, we will going to talk about the mostly chromosomal mutation. Alright, why chromosomal mutation? Because this particular section talks about what will happen if you have extra number of chromosome or lacking number of missing number of chromosome or missing chromosome in your body. All right, so we call that chromosomal mutation as autosomal aneuploidy. So what does it mean? It tells us that this is the condition of having abnormal number of chromosomes. So autosomal aneuploidy is part of chromosomal mutation. Now it can be a monosomy or a trisomy. So what do you mean by trisomy? The chromosomes have three pairs, or rather as extra one chromosome. So instead of having a pair, it becomes a trio. Alright, so next one, the monosomy is missing one chromosome. So instead of having homologous chromosome that we have right here, so one chromosome is missing on the process, so there's only one chromosome on that particular set. Okay, so that's the autosomal aneuploidy. Now, commonly cause of autosomal aneuploidy is error in Nondisjunction. So this is the failure of the homologous chromosome. So when we talk about homologous chromosome, these are chromosome of same shape, same gene sequence, and also same size. Alright, so same pattern as well. Alright, if this homologous chromosome didn't separate properly during meiosis, the effect will be the number of chromosome will be altered in the process. So if the number of chromosome is altered in the process, therefore the physical appearance and also the functioning, the normal body functioning of the organism will also be compromised. Alright, so during meiosis, the homologous chromosome should be separating properly. So this is one cause of autosomal aneuploidy. And other causes of autosomal aneuploidy is radiation or even chemical exposure. Alright, so this is one. Okay, so there are two ways that non-disjunction can happen. So in the first place, we have normal separation that we have right here, but the non-disjunction happens in the meiosis too. So now this chromosome should be separated in a normal process, but in the non-disjunction, it didn't separate, so leaving one cell missing, having a missing chromosome. So instead of having 23, it is now 22. And this one, instead of having 23, it has extra chromosome which can cause trisomy. What happens here is it becomes 24. Now the one that we have right here, instead of having 23, it is missing one, so it can cause monosomy. Alright, so and the other one is, it should be like this. It should divide the way that we have right this on this picture. Alright, so in this side that we have right here. Now, non-disjunction can happen on meiosis 1 as well. So if the parent cells or if the parent chromosome didn't divide in the process, therefore the two cells that will, or the daughter cells that will produce, is one with extra number of chromosome and the other one doesn't have any or missing chromosome at all. So the if the meiosis 2 is normal, so basically it will affect the total number of chromosome on the daughter cells in the meiosis 2. Alright, so as you can see here, instead of 23, it becomes 24. And instead of 23 here, there is missing a chromosome here, it becomes 22. So technically, this is what non-disjunction during meiosis is all about. Alright, next one. One example of autosomal aneuploidy is what we have right here is the trisomy 21. So why trisomy 21? The 21 is the number of chromosome of it happens on the 21st chromosome. So if this is in human, this is you just look for the 21st chromosome, and from the word trisomy, the problem here is one of the chromosome in number 21 has an extra number of chromosomes. So we have an extra chromosome here on 21, so instead of having 2 only, we have now 3. Alright, so what is this one? this is this is a very familiar disease called the Down syndrome. Alright, so Down syndrome. Alright, so as you can see here, these are the physical appearance that you can take note upon a person with Down syndrome. Alright, so basically, maternal age is somehow affects the frequency of producing or the chances of having an offspring with Down syndrome. So as the parent ages or as the parent age increases, basically the chances of having or giving birth on an individual with Down syndrome is also increased. So this is directly proportional. All right. Next one. Next one is the trisomy 18. So what happens here is instead of having it in number 21, we have an extra chromosome on the 18th place of this karyotype that we have right here. All right. So as you can see here, we have 3 instead of 2. All right. So what we have right here is the trisomy 13 is the Edwards, all right, or Edwards syndrome. All right, so what you see here are these are the characteristics of a person who has an Edward syndrome. All right, so Edwards syndrome. All right, next one is trisomy 13. So what we have right here in the trisomy 13 is on the 13th chromosome, there is an extra chromosome that we have instead of having 2. So on the 13th. All right, so okay, so we call this one as the Patou syndrome. All right, so Patou. Right, so Patou syndrome. So these are the characteristic or the physical appearance of a person who has a Patou syndrome. Alright, so there is a misplaced or the development of the eyes is seen here, or the undeveloped eyes, or there's an undeveloped eye and also undeveloped mouth that is seen here on this organ or on this individual. So alright, so that is the effect of having an extra chromosome on number 13. Alright, next one. Alright, so if there's a trisomy, there's a monosomy. So we call this one Turner syndrome because there is a missing chromosome. Alright, instead of having 46, so we have 45 chromosome over here. The one that is affected here is the sex chromosome, which is the X and Y. Now, we cannot identify if this is a male or a female because it only has one chromosome here, and that is X. There's no Y or there is no X chromosome that is present here. So what will happen here is completely the organism will look differently from a normal one. So the Turner syndrome organism or the Turner syndrome, the person who has Turner syndrome has this set of characteristics that you can take note of. Alright, so alright, so these are the characteristics of an organism who has Turner syndrome. Alright, next one. Alright, so again, alright, there is a trisomy case here. So this is what they call the Klinefelter syndrome, and these are the characteristics of a person who has a Klinefelter syndrome. So what happened here is on the 23rd chromosome, which is the sex chromosome, basically we only have X and Y, XX or XY. In this case, it has an extra chromosome that we have right here, which is instead of XY, we have XXY. All right, so meaning to say the person can be classified as a male or a female at the same time. All right, so this is what they call Klinefelter's syndrome. All right, so they are this organism or this individual are sterile, they cannot reproduce. The same way with Down syndrome, the Edwards syndrome, and the Patou syndrome. All right, next one. Alright, so basically, those are the autosomal aneuploidies that are seen in humans, or cases that are found in humans. Alright, so other kind of mutation is albinism. So albinism is the inability of the skin to see or to give off melanin. So technically the person doesn't have melanin on its skin, so the person's skin is colored white. Alright, include not only the skin but the eyes itself is also affected. Yan, so instead of the color of eyes, or instead the eye color is somehow black or brown, it is colored red. or it is color red. It indicates that if it is color red, the presence of melanin is less in that particular region of its body. So that is albinism. Some animals also exhibits albinism, like in zebra. In zebras, normally zebras have stripes. So albino zebra, basically these are zebras that are colored white. Alright, so in some crocodiles, there are white crocodiles, there are white reindeer, something like that. So these are some cases of what they call albinism. Alright, so if you want to know more about the genetic disorder, actually there are a lot of genetic disorders that we can discuss here, but since we are limited in this particular video, so you can use this link to look for some genetic disorders if you are interested. Alright, so this is a person with albinism. So as you can see, this person is completely different to the other people around him. Alright, so this colored one. Alright, so okay, so next one. Alright, so basically that is mutation. Alright, so at this video you have learned about the two types or two kinds of, or what is mutation all about. Next, the two kinds of mutation, we have the gene and then chromosomal mutation. So under the gene mutation, it is there are two classifications of it. We have hereditary and acquired mutation. Alright, so also in this video we discuss deletion, inversion, insertion, repeated expansion, duplication, missense mutation, frameshift mutation, nonsense mutation. And also, don't forget that we discussed how the number of chromosome or extra number of chromosome affects the physical, and not only the physical appearance, but the normal function of the body of the particular person who has an extra number of chromosome, which is called the autosomal aneuploidy. Alright, so with that, I hope you have learned something about this video. Now, if you have comments and suggestions, don't forget to write it down in the comment section below this video. Alright, so special shout out to my students here in Muntinlupa Science High School. Alright, so with that, see you again on the next video, and goodbye. Always smile, alright, and have a nice day, everyone.